El ICA le da la bienvenida a este evento y agradece su participación. Estas son algunas recomendaciones para mejorar la calidad de la videoconferencia. Habilite su micrófono solo en el momento que le den la palabra. Es importante mantenerlo en silencio cuando otra persona está hablando. Por favor, cámara en todo momento, para que otras personas lo puedan ver. Si necesita atender algo personal, puede apagarla y encenderla al volver. Agradecemos por favor poner en silencio sus teléfonos celulares para evitar alguna interrupción en la videoconferencia. Recomendamos cuide su postura y su posición frente al computador. Para esta reunión contaremos con interpretación simultánea. Es muy importante que cuando ingrese al enlace de Zoom del idioma de su preferencia, debe hablar en ese idioma. Por ejemplo, si ingreso al enlace de español, debe hablar en español en todo momento. Al finalizar el video, les pedimos el favor de hacer silencio durante 30 segundos. Una vez finalizado ese tiempo, el moderador o conductor puede iniciar la reunión. Muchas gracias por su atención.
Okay, well, on behalf of my colleagues from the Prosa Norte Plant Health Task Force, Sergio Paulo from Canada and Jose Lopez Arroyo from Mexico, I would like to welcome everyone to the Plant Health Task Force now third annual virtual webinar series. This was a two-day series that started on Tuesday. Today, we will be focused on mitigating the effects brought on by climate change on grapes. Tuesday was focused on wheat, and we had some great presentations, and so I'm really looking forward to today's presentations. And now it is my great pleasure to introduce Dr. Stephen Kappas, the current president of the Prosa Norte Board of Directors, to give some opening remarks. Dr. Kappas?
Thank you, Dr. Winmar. And it's my pleasure and honor to welcome all of you to this Prosa Norte Plant Health Task Force webinar. As Dr. Winmar indicated, it's on grapes, and in particular, the impact of climate on the plant health of grapes. The mission of the Prosa Norte Plant Health Task Force is to share knowledge to promote sustainable management of insects, diseases, nematodes, and weeds that negatively affect crop production in North America. Grapes are an important commodity to all three countries, both for fresh market and wine production. Since Canada produces only 2,750 metric tons of grapes in 4 provinces, they must import from the US and Mexico to meet the consumer's demand. Mexico is the largest producer of fresh grapes, and they ship over 200,000 metric tons of grapes to the United States and 25,000 tons of grapes to Canada. US will also export 93,000 tons of fresh grapes to Canada. Grapevines, whether grown for fresh market or wine, is impacted by the climate factors such as increased heat, drought, fires, increases in pests and diseases, and unpredictable and unexpected frosts and freezes that not only impact yields, but also fruit quality. A 2 degrees Celsius increase in temperature could shrink the regions that are suitable for wine production by 56%. That's amazing that it would have that large impact. So today you're going to hear an excellent list of speakers from all three countries give presentations on research activities related to mitigating the effects on grape production brought on by climate change. And I want to thank Dr. Winmar and his co-chairs and ICA for organizing this event. Turn it back over to you, Dr. Winmar.
Thank you very much, Dr. Kappas, for taking the time to open our webinar series. And before we begin, I would really like to thank Daniela St. Victor and Gloria Ramirez from IICA for all of their help in putting this together, and also for our interpreters, Cynthia Diaz and Luciana Pavez, who are doing an excellent job in interpreting and translating so that we can all understand the presentations. And so now for our first presentations, I would like to turn it over to Jose. Jose?
Muchas gracias, Tim. Es un placer
Thank you
estar
so much, Tim. It's a pleasure to be here today. It's a second seminar this year, and warm greetings to all of you, wherever you may be. I am Jose Isabel Lopez Arroyo, and I proudly represent INIFAP in the Plant Health Task Force at Procinorte. Today, we will begin the seminar with a presentation by the Mexican bloc. And firstly, we will have Dr. Hector Gonzalez Hernandez. Dr. Gonzalez has a PhD from Hawaii University. He is currently working full-time as a professor and researcher at the postgraduate college. Throughout his professional career, he has been in this same college. Last year, Dr. Gonzalez received the National Award for Plant Health because of his scientific research. This is a very prestigious award in our country. And today, Dr. Gonzalez will be presenting incidents of the vine mealybug in the main grape regions from Northwest Mexico. Dr. Gonzalez, you have the floor.
Sí, José Isabel, muchas gracias.
Thank you, Jose Isabel. Thank you. And thank you to the organizers as well. I really appreciate the opportunity for sharing some of the experiences we have with vine mealybug.
Muy buenos días. Solo para preguntar si...
Just to check if you can see my screen. Is it showing full screen? Yes, we see it.
Muy bien.
Thank you.
Buenos días a todos.
Thank you. And once again, good morning, everyone. We already heard what I will be talking about, the title of my presentation. And to share some of the content, we will be covering some of the species of vine mealybugs, the economic importance, financial importance that this species has, particularly the Planococcus ficus. We will also talk about the origin and distribution. That's very important, especially for biological control programs. And I will also refer to the biological characteristics and the types of damage. I will also share some information on how the distribution and behavior has been seasonally of the mealybug in the state of Sonora, which is the main grape producer in Mexico. It has about 20,000 hectares of the total 35,000 or almost 40,000 hectares we have in all of Mexico, so just 20,000 in Sonora. I will also refer to the case of Baja California and the incidence of the mealybug there. Baja California is important also for grape production in Mexico, especially because it concentrates the main producing region for wine. So it has about 5,000 hectares there. And then I will also focus on some of the effects that climate change can have in this particular area, and more specifically in Ensenada and the Guadalupe Valley, where we have the main production of this grape. So I'll talk about the levels of incidence, and I will refer some of the information of the project we currently have with budget from the General Directorate of Plant Health and the National Phytosanitary Council, and then some conclusions. So just briefly, the importance of mealybug overall in wine production internationally. We have data sources and much information showing us that about 30 species of mealybugs have been associated to wine and grapevines. Some of the examples are the species we see listed here. Some have very important financial importance or very prominent financial importance, such as Dismicoccus brevipes. And some of these species are cosmopolitan in their distribution. They can be found everywhere. They affect different crops, fruits, and even ornamental plants. Then we have Faurecia virgata and Macrolenicoccus hirsutus, the pink hibiscus bug, then other species like Paracoccus and Phenacoccus. We have two species of Planococcus, the most important for wine and vineyards, Citri and Ficus, and several species of the Pseudococcus genre, the Longispinus, the Maritimus, and the gray mealybug, Viborni. So here we have some of the species that you can find mainly in the North American productive regions, like in Mexico. Just to mention a few, the Maritimus, Pseudococcus maritimus, greatly distributed towards the south of the United States and the north of Mexico. And the greatest incidences, I've said, for Sonora and Mexico, according to our records, starting in about the 2000s, the wine mealybug Planococcus ficus. We also see the Longispinus, and there are some records of Pseudococcus viburni, especially in the south of the US and California. In Mexico, it's very, very restricted in its distribution, and it has not been detected in grapes. So we can basically say that in Mexico, for grapes and species that are of financial importance are those two. Now, what's the importance of this mealybug internationally speaking? It's a considerable pest in South America. You find it in Argentina, California, Baja California, and Sonora in Mexico. It's been found in Peru, South Africa. And what we've detected in Sonora in 2001, for example, about 150 hectares with approximate losses of $2 million. So the impact this can have, imagine in some of the cultivars of grape for Merlot, Malbec, and Cabernet Sauvignon, when they are infested by this mealybug, there's high infection of the fungus Aspergillus nigri section, which has a potential risk of contaminating the whole wine production chain. As I said, it's important to understand how this species came to be, the origin. It is of Mediterranean origin, and it has been determined that the populations found in Turkey, Tunisia, and several regions of Europe are related to the populations we find in South America, Argentina, and those that we find in Israel and Egypt are actually the ones that relate to the populations found of vine mealybug in the southern United States and northern Mexico.
También es importante mencionar las características.
We should also mention the biological characteristics of this species. Why is this pest so important? Wherever you may find it, it will lead to great loss. It has a high rate of reproduction. The female can lay up to 250 eggs, and there can be even 6 generations per year, with some overlaying generations. So throughout the year, you can find the different stages of development or adults of this species. What we found in the Guadalupe Valley is that this species reduces the number of leaves, so it has a direct impact. There's less foliage, the height of the plant is decreased, and one of the favorite varieties, if we want to call it that, an important variety, Nebbiolo, is less tolerant to the presence of this bug. Other biological features, and it's basically found throughout the plant. You can find it in the roots, stems, and bunches. And what determines where it is presented can relate to environmental factors like extreme temperatures under 15 degrees, for example, or above 37. So it has to find areas of shelter if it has extreme temperatures, and then that makes it difficult to find it in the stem or the root or under the bark. And it's really difficult to manage because it seeks out all parts of the plant. And where it attacks will also depend on the phenology of the crop. And finally, the greatest damage comes when they start infesting the bunches. They are contaminated. There's basically, you have to discard the whole bunch. You cannot use it for direct consumption unless even for wine production. We have a table here that shows the length of each cycle of development. Most importantly, we have the full development cycle. It takes 24 days. Longevity, it's more or less the same time.
Es muy importante.
And then fertility is very important. I've mentioned this before. On average, you can have 250 eggs, but you can find even 450 eggs by a single female. Other characteristics, particularly for this species of mealybug, is that it attacks more than 36 species and different botanical families. And some of these plants are financially important. They're palm trees, ornamental plants, you have other fruit trees, vegetables. It really consumes all types of plants. And the direct damage you see in the grapes is just one of them. As I said again, you can find it practically in all continents, and it is distributed everywhere. It has a higher incidence in Europe. There's also an important mutualistic relationship with several species of ants. For Baja California particularly, we have found certain species of ants that are dominant in some cases, like the Formica purpulosa and the Neptela humile, which is the Argentinian ant, and they are broadly distributed. And how does that mutualism work? Some of you might already relate it to other systems where you find sucking bugs and others. So in the case of ants and mealybugs, first, you see that the ant exploits the resource, the honey that the mealybug excretes. And if it wants to protect that resource, of course, the ant will need to provide some kind of protection to the bug. If it cleans it out, then the immature ones will have direct access to any area around the substrate. But if the ant does not take that honey, that sap away, it could be a factor of mortality. So the ant needs to really protect that resource, and it protects the mealybugs from its natural enemies. So there's that mutualism there. Both individuals benefit from that association. And in this case, for the vine mealybug, it protects it from wasps and other predators. The greatest impact is on parasitoids. Here we see a very specific species, Anachirus. There's two species that are related internationally, Pseudocoxi and Vladimiri. and they are even used in biological control programs. And there's a commercial production of these. Now, what have we found lately in Baja California and Ensenada, particularly the Guadalupe Valley? Extreme temperatures make the mealybugs seek protection under the bark. So under 15, above 37, the bug has to remain under the bark to shelter or even in the roots. And so it makes it difficult to manage. It's difficult to access first for natural enemies and sometimes even ants as the most important action in managing mealybugs internationally. So biological control is a very successful tactic, but if you have integrated management, the use of selective insecticides, you would have to have systemic insecticides because if it's under the bark, if you apply a contact product, it will not touch them. So that's the limitation. What we've also seen, and producers have observed and told us, the greater incidence of this bug happens when there's vegetation growth, when the bunches start growing, and in wintertime, they are only found in the roots. So that is changing because as we have observed, again, you can find it any time of the year, and any time of year you will find them under the bark, in the roots, and even more so if you have ant activity. What other types of indirect damage do we find when we have the vine mealybug? It's a vector of a virus of leaf roll. So being the vector, then it can lead to this virus GLRAV3. 3 refers to the subspecies. And this is the one that is most closely related to the presence of mealybug, Planococcus ficus, in the grapevines. There are other mealybugs, other species that I've mentioned before that are also associated to leaf roll. Practically all species of mealybug can be vectors of that virus. Now, what have we seen in the seasonal distribution of the mealybug? In Sonora, for example, when we first saw it in Mexico as an important pest, and this was around 2001, 2002, here we see the graph showing that the greater peaks are mid-year, and then with an increase in temperatures, you have vegetation growth about March, April, and then the bunches are attacked halfway through the year, about June. And what you see is there's presence of adults and other stages of development. And as I said, this bug is related to the phenology of the crop, so greater incidence in the middle part of the year when you start having bunches. It can also be found in other parts of the year in the leaves, but mainly there. And the damage is when it starts attacking the grape bunches. Some more information that we've had recently, about 2015 to 2018, we see levels where producers have already started managing and so reduce the incidence of the bug to very low levels, really.
Productoras, por ejemplo, de Sonora
In certain regions like Sonora and the Hermosillo coast have lowered that. Now let's look at the effect of climate change, the effects that climate change could have on the incidence of this pest, of Planococcus ficus in the area of Ensenada. Here we see a historical graph of temperatures from 1979 to 2021. And you see there is an increase, a gradual increase. And it could be reflected also in the bug's behavior. It could even favor, in certain conditions, the development of the population. We know that optimum temperatures of 27 degrees can occur in basically all months of the year. Now here, in the bars, we see the darker colors when there are important changes in average prevalent temperatures in the region. And the same for precipitation. So we see here that in a year, temperature changes and it can increase. And this is all related to phenology. When can we have greater incidence of the mealybug under the ground? That's basically the first 6 months of the year. And then when you have the mealybug in the stem or in the middle top part of the tree, it's the mid part of the year, and then in the leaves when you have vegetative growth, and in the bunches also to the second part of the year. This is the incidence and what we've observed in the Ensenada case. The first detection was in 2014 with 80 hectares in that year, and gradually it has increased. In 2015 to 2021, you've almost doubled this surface, the area covered. What you see in red here does not necessarily mean that it's a high level of incidence, but if we look at what our sampling committee does in their campaigns, the Plant Health Committee, regionally, we have few areas that are strongly infected, or you see 1 to 10 bugs per plant. That's how we look at strong, and medium is 10 to 20. And those are the thresholds that can be considered to suggest management strategies. This is what we've seen in direct sampling in plants, monthly, 2021, 2022, populations remain at low levels, we can say, manageable.
So this is what we are currently seeing with the project. As I commented, this shows the management strategies being used in this region, this wine growing area, grapevine growing area. Basically, they use insecticides. Although they are selective, they still have an impact on the fauna. Basically, I'm talking about certain groups of monocotyledons, although they are using fatty acids synthesis system, but it has its limitations. And if pyrotetramac is contact, then they do not debark, well, the application will be not be as efficient. We have also seen that there is a positive fauna there, and we have detected the activity of Anagerus vladimiri under the bark. under some conditions. That is, there are regulations by natural enemies. There are all other predatory species. Mutorani puntacieri in Brazil, they use that. So that is what they are using also in Baja California. So what ant species have we been detected? Well, Formica purpulosa is a type of ant that has it is basically in the deserts in northern Mexico. This is an Argentine ant. And for conclusions, this type of vine mealybug has greater incidence in the vineyards in Baja California. The presence of this under the bark and in the trunks basically occurs all year long, perhaps due to the difference in very low temperatures or very high temperatures, both in winter as well in spring and summer, and the continuous association with different ant species. Now, the project is currently aiming at looking for the best ant management. If we can manage the ants, the incidence of the vine mealybug will be less. And with this, I have finished my presentation. Thank you. Go ahead. Thank you, Dr. Gonzalez. If you have any question, please do not hesitate and raise your hand or write down your questions in the chat. And while someone dares ask Dr. Gonzalez, perhaps Dr. Gonzalez could talk about the strategies they are proposing to manage ants. Well, it really depends on the ant species, the bait that should be made, and in stations where we can ensure that only the ant will eat the bait, because the bait can be made with some insecticide product at a very low rate, and even with boric acid. We have seen it has a good effect, so it could be liquid formulations or solid formulations, depending on the species. For example, for Formica purpulosa, they have been using a solid bait with boric acid, in fact. It gives time to the ant to go to the to feed the rest of the animals in the colony, and with this it can have a bigger impact. It can, if we can reduce the activity of the ants, then the activity of natural enemies will increase. Well, thank you very much. We have a question by Dr. Witter. He's asking how common is the Aspergillus niger contamination? No, in Mexico, no, we have not detected that as such. But there is a risk that if the grape that has been attacked by the vine mealybug is used, there could be problems in the wine production chain. And these ochratoxins are toxic for human beings, obviously. Oh, there's quality testing to determine the quality of wine, but anyway, it is still latent. If you are not careful in selecting the grape, there could be problems with the incidence of this fungus. Thank you very much, Dr. Gonzalez. We are very grateful for your participation. Very interesting presentation. Thank you very much. Thank you for your attention, everyone. We will continue then with our seminar with a presentation by Dr. Gerardo Martinez Diaz. Dr. Gerardo is a researcher at INIPAP. He's a colleague of mine here. He has more than 40 years of experience working at INIFAP. And he has a PhD in 1997 from the University of Arizona. He is an expert in wine growing, grape growing, excuse me. And today, Dr. Martinez will present managing the grapevine trunk disease in face of climate change. Go ahead. Hello. Yes, we can see this. Yes, we can. Okay, fine. Thank you. Well, thank you for inviting me. Particularly, I thank Jose. And I will now speak about the grapevine trunk disease. I will start with this slide showing the grape growing areas basically for wine, but in practice, these same regions also produce grapes for a fresh consumption or to make raisins. So this shows the importance of the grapevine in each one of the regions. And we can see it according to the size of the circle. In the case of Mexico, as you can see here on the map, well, our circle is very small. However, it is important above all for the production of grapes for fresh consumption, because a good part of the production is for export purposes. And this means getting a lot of currency and foreign currency for the country. At a national level, the map here shows you the distribution of the production areas in Mexico, but in practice, as the previous speaker mentioned, grapes are grown basically in northwestern Mexico. 85% of the grapes for wine, table wine, are grown in this area of Baja California, and more than 90% of the grapes for fresh consumption are grown in this region, also in Sonora. So that means that for the remainder of the country, perhaps maybe 10–15% of the surface is used for production in those other areas. The map here shows the location of Sonora, where the grapes are grown for fresh consumption. In this area, this is a desert climate. So in summer, we have extreme temperatures, very similar to those, for example, in Arizona. Maximum temperatures are 50 degrees C, and minimum could be up to 2 below, only in this region. Precipitation or rainfall is about 140 millimeters throughout the year. 56% of that amount is concentrated between July and September. In this same area I was commenting, soils are alkaline with a pH of 7.2 to 7.8, and we have mean texture, although there's about 20% with the heavy texture soils and 17% with light texture. So up in the highlands, the weather changes a bit. So in this region, we can grow wine grapes, but this has not been explored much in the region yet. Now, as to diseases that have been detected in Sonora, well, it is very difficult to specify how many diseases attack the grapevines, but it is calculated that there are about 60 different viruses, 2 of which have been reported in Sonora only. As to bacteria, there are 3 worldwide and 2 have been reported in Sonora. For nematodes, about 15 worldwide 10 of them in Sonora. And as to fungi, they are calculated at 130, but for the trunk disease, it is, some consider there are up to 140, but using the 130 figure in Sonora, only 23 have been reported. And this is in a brochure published by a master from the University of Sonora, Mr. Avila. As to the trunk diseases, here we can see the table. We have the disease called by Lipodothermobrae. We have this disease caused by Tifa, and its presence has been reported caused by Domicifolia, and the Blackfoot by Cylindocarpo and Phomorphis viticola. Allow me to mention there are some diseases caused by these agents that are not on the list of the main trunk diseases. These are Ganoderma stearium, but we also have these Fusarium and these others here listed here. There could also be others, but we still need much to study in this great diversity of trunk fungi. This is a vineyard affected by this type of diseases. So as time passes and the disease gets stronger in the plant, notice the poor growth. Sprouts are much fewer. And lastly, there's a dieback until we see them just completely die out, as we can see on these grapevines. Over time, many of them die, and their productivity decreases until the vineyard ceases to be economically profitable. And this slide shows another vineyard infested by these trunk diseases, and they ended up having to just eliminate them. When we examine the fungi, the prevalent one is the one that produces this type of spores. And these are grouped in the Botrosporiatic family. The symptoms, well, basically correspond to these here, these images. Botrys aerea or Tifa. But there are other types of symptoms also. Okay, trunk diseases have been studied over time by different researchers. The oldest reference in the case of Mexico, at least, is that by Tellus in 1979, reporting the presence of Ganoderma. Then in the Institute bulletins, INIAP, there is reference of Utipalata, which was distributed in different parts of Mexico. Then in 1991, Leavitt examined Mexico and found this Botrosporiaria, and as well as Utipa. Researchers from University of Davis, Urbes Torres et al., studied the area also and came across Lassi diploia and the diploia. More recently in Baja California, there was presence of these two fungi causing the Petri disease were detected in the area. And even more recently, Lopez et al. From the University of Sonora did some work where he published the existence of C. diploia tebromai. And there's another recent paper from Baja California also, noting the presence of La C. diploia brasilensis. This is a much more recent paper exploring how to better manage these fungi. Pruning in our region is done late December, early January. In these months, rainfall is very scarce, very limited, basically 20 millimeters throughout 3 whole months, January, February, and March. In the case of regions where the weather is more Mediterranean style, rainfall is more in the winter season, and that is when these are pruned. So in these regions, due to the presence of rainfall, spores are produced that will be carried by wind or by any other environmental or human condition and will carry the spores to the cutting areas. So this apparently is how the fungi spread in these areas. There are studies to see the number of spores that are found in the environment at different times of the year. These studies, however, do not exist for our region. For our region, rainfall is concentrated in the summer period between June and August. As you can see, rainfall is about 57 millimeters, 3 times more than in the winter. So we could think that the way this pathogen or these pathogens are spreading could be different from that in Mediterranean climates. However, this has not yet been studied to determine how these diseases are propagating. One of the first studies carried out here more recently, and these have been published in this technical report of the wine growing seminar we hold in the region every year, is that first a grapevine lot was selected that had the trunk disease symptoms. The fungus was isolated, and then through molecular techniques, they concluded or identified that this was Lidiproia temobrae, the fungus that we have mentioned several times as identified by previous researchers. And after that, there was another study carried out analyzing the susceptibility of different cultivars in the region. There are about 16 different varieties to produce table grapes. Some of these varieties are patented, others are free. The interesting part of this paper was that although this followed a methodology to identify short-term susceptibility, they found that there was a difference between different cultivars. They formed 5 statistical groups, 3 of which gave similar results to a control where they were not infected by the fungus. So it is very likely that some of the grape cultivars are more tolerant than others. At the same time, we have carried out some other studies with Vitis arizonica. Vitis arizonica is wild grape that, as you can see here, can grow along with native vegetation, climbing up these trees to heights up to 20 meters. And aside from working with this, we have selected Vitis vinifera for some other studies. So over these years, in these 20-liter buckets, They have made some studies for the strain of Ericepa teobrae that was isolated in the field, was raised under laboratory conditions, and was used for this type of assay where we make a cut in the tissue until we reach the wood, and then we proceed to infect or apply whatever products we want to evaluate to see what happens over time. In this case, the evaluations are no longer 15 days after infection, as is the case with short-term studies. In this case, instead, we have waited up to 4 or 5 months to see what is happening with the damage. Through the different studies, what we have seen is that the fungus is capable of entering not only through the prune wound, but through other types of wounds caused commonly in intensive handling of grape for fresh consumption. In grapevines destined for this type of production, where they remove the leaves, they remove the sprouts, they make the rings, and they prune in the summer. So this intensive handling makes the grapevines be exposed to the arrival of spores or mycelia that could be that could arise through this intensive handling and infect other plants or in the different parts of one same plant. So the irrigation system is always through drip irrigation. And some of the growers put the drip hose, as you can see here in the back, several centimeters above the ground. So oftentimes the trunk gets wet in this region, and that moisture or humidity makes causes the proliferation of the fungi and the release of spores. We have also found that spore production can occur year-round. All we need is high humidity for this to happen. So what do we suggest in order to control these diseases? Well, most common is to obtain a pathogen-free plants. Quite difficult, right? Because the materials are sometimes already infected from origin, and there is no control really for this type of diseases. For other diseases that there is, but for the trunk rot, the disease, there is really no legal control to prevent materials that arrive or that are being reproducing are free of the trunk diseases. And the other thing is, well, this is planting in sections on a new ground, for example, which could be done in this region because there is so much land available, and this can be done. However, some growers prefer to destroy a vineyard that is no longer productive, and right on that same soil, they plant again. And this is we do not necessarily suggest. As to chemical and biological controls, I would like to show you this slide here, where we show a program to handle the different diseases. And we have botrytis, nematodes. Basically, it says when the management occurs. In the case of trunk diseases, we suggest that after pruning, an action be taken, but then also take action throughout the entire grapevine growth. There is a fungicide we have studied. Although it has been prohibited in other countries, it is still allowed here in Mexico. That is benlate. This fungicide does work following this method that I already explained. And the same occurs with myclobutanil. So we suggest that after pruning, we apply this to reduce the infection, at least for Seriploia dermomae. We do not know what can happen with the other types of fungi because there are several others in the region. So throughout this period, and this not only applies to trunk diseases, but there's a frequent spraying of trichoderma. This could also help, but we are not certain as to how much control is really obtained, because following the methodology that I mentioned, at least for this strains used in this region, we have not found that the Seriploia infection is controlled. Nevertheless, there are many reports in Europe as well as in California mentioning that trichoderma has a potential to reduce the infection of wood by trunk diseases. Possibly what is needed is perhaps to explore other species or other races. Aside from this, we must consider that not only workers, the day workers or technical experts are spreading the fungi through the different practices in the vineyard, but pests are doing part of the job too. There is a publication here finding that some insects that affect the grapevines carry these insects, and this could happen with insects like this, the vine mealybug that we have been talking about. Now, what to do with all the wastes after pruning? Before, all of this used to be burned, but the residues later were crushed, and then everything, the remnants would end up under the grapevines, and this is still a practice. The fungi are spreading even further. So we suggest that instead of burning this, use composting instead. And instead of the chips, put them on the ground.
Over time, the grapevines produce less until the damage is irreparable. However, if the fungus has not yet reached the basal areas of the trunk, you can still recover the grapevine. Here we see an example of a vineyard that had been producing for 15 years. It saw its capacity reduced, and the option was to cut off about 40 to 35 centimeters in height, and then it started growing again. And so you can see that here. And this grapevine is still highly productive currently. So wood diseases are still a threat for wine growing in Mexico. We need more studies to continue identifying and looking at biological control methods. What perspectives do we have? Well, again, we have to continue identifying species. We need to see how we can control the disease legally and incorporate new strategies for monitoring and biological control, as I said, especially in the case of this fungus that we've been studying, the trichoderma. We did not see the results we wanted, so we need to find other species, other organisms that are not related to trichoderma as well. We also have to continue studying the interaction of water stress and susceptibility to these fungi. Water stress will continue. We will see plants that are increasingly stressed because water will be an element that reduces over time if we consider the climate situation. And also in these regions, the water that is used for irrigation comes from the subsoil, and these phreatic aquifers are decreasing also. I wish to thank Juan Pedro Lopez. He's a researcher from Sonora University. He's been working with us on the project, as well as Aurora Fontes from INIFAP, Ms. Rufina from CICESE, who has provided some of the species for studies, and also to Akif Escalin from UC Davis. We've interacted with him to receive suggestions. Thank you very much. Thank you, Doctor. We have a question in the chat box related to the availability of rainfall and moisture. So do you see an increase in temperature or rainfall? Would this lead to greater incidence of these diseases? Could you please comment? Yes. In this region, thankfully, and I think that's why we have not seen any other diseases, we have very low relative humidity. If we compare it to the number of diseases you find in other regions in Mexico or the United States towards the east, we have a series of diseases that have financial importance, and some have adapted to the low humidity conditions, like the ones I'm showing, and the trunk diseases we've seen. And that's where we see the damage. Thank you very much. We have other questions in the chat box. Hopefully you can answer them yourself directly. Thank you. Very interesting presentation. Thank you. It was a pleasure. And now we give the floor to Sergio Paulo, who will be moderating the next session, which is presentations by colleagues from Canada. Thank you.
Thank you, Jose, and thank you, Dr. Gonzalez Hernandez and Dr. Martinez Diaz for the great presentations. So I hope everybody can hear me okay. My name is Sergio Paulo. I'm one of the 3 members of the Pro-Norte Plant Health Task Force, and I'm currently a director with Agriculture and Agri-Food Canada within the Science and Technology branch. As our next speaker, I'd like to introduce Dr. Mehdi Sharifi, who is a researcher at Agriculture and Agri-Food Canada's Summerland Research and Development Center in British Columbia's Okanagan region. His research activities are focused on enhancing the environmental sustainability and resilience of perennial horticultural crops through ground vegetation management and the use of organic soil amendments or help promoting natural products. Dr. Sharifi will be giving us a presentation on improving vineyards' economic and environmental sustainability using alternative management practices. Dr. Sharifi, the virtual floor is yours.
Good day, everyone. Thank you so much for the organizers of the workshop. And thank you, Sergio, for the introduction and inviting me to talk at this workshop. So as Sergio mentioned, the title of the presentation is Improving Sustainability by Using Alternative Management Practices. We know that we're dealing with several climate change issues. Canada can expect more extreme weather events, higher than average temperature, changes in the precipitation, and which increase the risk of the drought and the floods. The picture that you see here is a picture of a field last November that flooded with the water in British Columbia, Canada. So agriculture sector must become more resilient and able to adapt with the climate change scenario and threats without relying more on the external inputs. So we need to look for ways to reduce greenhouse gas emissions and increase carbon sequestration on the vineyards. So that means a change in the paradigms and change towards the sustainable agriculture. We need to build resilience in our agricultural system, including vineyards. And this change can be done through in different ways. We need to enhance the yield stability and overall plant health. By yield stability, I mean that the changes in the yield over year as a result of environmental stresses. And there are different ways of doing that or contributing to that, improving resilience. One is organic amendments. Organic amendments has long been used, and they have ability to buffer the soil environmental conditions, moisture and temperature, support the soil microbiome, not only on the topsoil, but also in the subsoil by reactivating the soil microbes. We can promote the soil health, use the product that promotes soil health, plant health, such as seaweed extract. We need to look into the ways to accelerate the soil carbon sequestration, such as biochar and cover crops or the mixture, and use alternative sources of the nutrients, such as compost and vermicompost. Also, alternative for the pest management, and one of the products that we use is the vermicompost tea. And of course, enhancing biodiversity, it's a very important component of the resilience agricultural system. And we can enhance the biodiversity by the cover cropping and reduce use of agrochemicals. There has been renewed interest in the soil organic amendments for several reasons. One of the reasons is the need for the alternative fertilization and the pest management, that these organic amendments can be used as mulch to prevent the weeds, and also can be used for controlling pests or soil-borne diseases such as nematodes. There worldwide pressure for restricting the use of the glyphosates, and these soil organic amendments or the mulches can be an alternative to the glyphosate. Also, in many regions there are yard waste and forestry byproducts that can be used, recycled and used as mulches. These organic amendments can improve soil health, and as a result can improve the soil yield stability and reliability, and that could be used as a method for the adaptation and mitigation of the climate change, as well as other alternative products such as vermicompost and vermicompost tea. Literature shows that the vermicompost can increase crop yield, supply nutrients to the grapes, enhance arbuscular mycorrhizal fungi colonization in the roots, and support or suppress the population of the nematodes, particularly parasitic root nematodes. Vermicompost tea can improve the grape vine growth and development, as well as fight with the diseases such as powdery mildew, which is a major disease in the vineyards, and botrytis. The next product is the seaweed extract. Has been a lot of research on recently on the seaweed extract. It can improve the plant establishment by promoting the root development. It can stretch the bunches and reduce the diseases by that. It can increase the set in the grapes and enhance overall wine health and yield. Can improve the color uniformity in the berries and enhance the cold hardiness, which is extremely important in the northern regions. And of course, the quality and also can reduce some reports on reducing the sour rot. So the region that we live here is in Canada, British Columbia. It's the western province of Canada. And in that region, we have the Okanagan Valley, as you can see, which is a semi-arid region with the average winter temperature of -1.4. Summers are warm, average of 19.4, but for a month or more, we get plus 35 degrees in this region, and annual precipitation is very low, so it's 300 millimeter per year. And most of the vineyards are irrigated, drip irrigation mainly. And this is a major grape or wine grape production region in Canada. A series of the experiments started from 5-6 years ago with the objective to promote climate change adaptation and mitigation in the vineyards. We use different practices such as mulch, compost, biochar, cover crop, vermicompost tea, and seaweed extract, and we monitor the effects on the soil carbon, nitrogen, and carbon and nitrogen cycling, as well as nutrient status and grape wine yield and yield quality. The first section is to look at the effect of these parameters. It is different practices or alternative practices on the enhanced yield stability, enhanced plant overall health, and accelerating soil carbon. Experiment one was on a wine block that Merlot variety on SO4 rootstock established in 2011, and treatment applied every other year. The treatment include control, so no application of any amendments. Second was mulch, 129 tons dry matter per hectare. Compost at 6.1 tons per hectare, and mulch plus compost, and all these applications were under wine. In 2018, we collected samples from different depth intervals, 0 to 15, 15 to 30, 30 to 45, and 45 to 60. And we did several measurements, particularly with focus on the carbon and nitrogen. So we look at the total carbon nitrogen. We look at the carbon and nitrogen mineralization, water-soluble forms of those. We look at the DNA of the organisms, and we look at the also bacterial and fungal carbon cycling gene abundance. So we wanted to see the effect of these different treatments on these parameters. The first thing is yield stability and reliability. So we look at the changes in the yield over time. And as you see in the left graph, we see the yield from 2013 to 2020. And what we hoping to do fighting climate change is that to have a more reliable yield. So the slope of the line, the lower the better. And we see that in the mulch treatments, as well as in the graph in the right. We see the variance and also the coefficient of variance with the yield with different treatments. This is average over the years, and we see that much less variation in the yields observed in the mulch and also compost plus mulch. So the reason could be buffering of the soil environment, supporting topsoil or subsoil soil microbiome, as well as supply of the nutrients. We look at the carbon effect on the application of these treatments on the carbon at different depths, and this is the total carbon, and see the maximum effect was on the surface, 0 to 30 centimeter. We look at the dissolved organic carbon, which we think is the mechanism that supply carbon to the soil organisms below 30 centimeters. And here we saw the significant differences in all depths up to 60 centimeters. So both mulch and mulch plus compost supply more soluble carbon to the depth than the control. And control was just like the regular practice, which is the herbicide applications. We look at the carbon fractions, and as you see, mulch and compost, as well as mulch, provided significantly higher reactive carbon or active carbon in the soil, which supplies more nitrogen to the nitrogen to the soil, which supplies more nitrogen to the crop. And we look at the respiration, and as you see, the second and third graphs are mulch and compost and mulch, and significantly higher respiration, which means the higher biological activity in these two treatments up to the 45 centimeter depth. Then this is the results of the enzymes or the function of the organisms. And again, we see significantly higher in mulch and mulch plus compost up to the depth of 30 centimeters, as well as the total extractable DNA has showed the same pattern. Second experiment, we focused on the alleyways. In our region, we have sandy soils with very low organic carbon and low water holding capacity. So we were hoping to improve that and improve the carbon sequestration in the vineyards. So we had the same block of the Merlot, and we started from 2017, continued to 2021. And treatments were controlled in the alleyways, biochar and compost, a 1-to-1 ratio at the rate of 22 tons dry matter per hectare. And then we have cover crops, and then we have the combination of the cover crops and the biochar compost mixture. We monitored the carbon over time in these years. And what we found is that you see in the right graph, so we plotted the carbon sequestration, and we see in different treatments, biochar plus compost alone or cover crops, they did not increase the carbon in the soil. But when we combine these two practices, it's protected the carbon and sequestrates significantly higher carbon in the soil, which is we measured 11 and 17% increase in these numbers in 0 to 15 centimeter. Also in 2020, so after several years, we started seeing the effect on the yield. So we had 32% increase in the yield in cover crop plus biochar and compost compared to the control. In the next experiments, we look at the alternative sources of the nutrients, also alternative pest management and carbon sequestration. We did an experiment with the vermicompost tea. We sprayed it on the plant, and we look at the frequency of the application as well as the concentration. And you see in the left graph is the effect on the berry size. So we had bi-weekly application or monthly application. So 4 application versus the 8 application per season. And also we look at the concentration. So we had the standard concentration of the vermicompost tea versus the diluted. And you see in the third column is the one that we got the smaller berry size that is usually preferable for the winemaking, and we got most of the time higher ratio of the skin to the inside. Again, we look at the TSS or soluble solids, and soluble solids concentration increased when we had 8 applications of the standard rate. And usually the appropriate, this is a Chardonnay variety. The optimum range is 17 to 24, and we see that with that type of applications, we got into that range. We also look at the titratable acidity, which we prefer at the range of 0.6 to 0.9 gram per liter, and same treatment shows the optimum range. We look at the vermicompost application under wine, as you see in the picture in the right. We had 3 different rates. We had 7 tons, 14 tons, and 28 tons dry weight basis per hectare. And you see in the graph that in 2020, we saw significant effects on the pruning weight, on the titratable acidity, as well as on the total soil carbon and nitrogen. So the optimum values for all these parameters measured at the 14 tons per hectare of the vermicompost application under the wine. And these 14 tons is based on the under wine area. So we saw that, especially in that line that you can see, that's the carbon. So significantly, we doubled the amount of carbon when we applied these products from 2018 to 2020. So in 4 years, we doubled the amount of carbon as well as the amount of nitrogen compared to the control. And then we start seeing better values for the titratable acidity, and then we got the minimum amount of the pruning weight in the zero nitrogen control. So we had a grower control versus a zero nitrogen control. In 2021, we started seeing the significant differences in the yield, and we saw that the 14 tons and 28 tons, they start performing the other treatments. 7 tons, we had deficiency in the nitrogen. So we see that when you using these kind of sustainable management practices, you need to plan for a long-term application and be patient. Again, looking at the yield, by boosting the soil health, we improve the yield and yield stability over time, and you see the 14 tons performed better and less variability among the years. And the last experiment was the seaweed extract, which has been done a lot of research recently on those, especially in California. Again, we look at the Chardonnay in 2020 and 2021. 2020, we look at we saw the significant effect on the berry size. We saw the also significant effect on titratable acidity, as well as in 2021, we saw the clusters, the number of the clusters, and crop yield increased when we applied at the rate of 2.56 times per growing season. So what we compared the treatments was 2.5 liter per hectare versus 5 liter, and 3 times application versus 6 sprays, and they all spray on the plant. And the best results obtained at 2.5 and 6 time applications, and the significant results observed in the years that we had the maximum environmental stresses. Again, cold hardiness, we saw some positive effect of the seaweed extract on the cold hardiness in the cold years. Again, similar pattern when we look at the yield stability, we saw that when we had 2.5, 6 time applications, we had more stable yields, and eventually over time we got slightly higher yields. So in summary, climate change adaptation and mitigation management practices in the vineyards, they require long-term investment. We saw The effect starts showing from the year 3 to 5. Surface applied organic amendment or mulches can be used as a tool for adaptation to the climate change through promoting yield stability, soil carbon and nitrogen cycling, and soil microbial activity and functional diversity in the vineyards. Alley cover cropping combined with the biochar accelerated the carbon sequestration in the vineyards. Foliar vermicompost tea application enhanced fruit quality and reduced the need for the powder mildew spray. So we had at years 2 spray in that block at the end of the season versus the grower that had 9 applications. Seaweed extract application enhanced grapevine resilience to the environmental stresses. With that, I would like to thank you, the several people who contributed to this research, including the students and technical staff, as well as funding sources from the BC Wine Grape, Agriculture Agri-Food Canada, and CGCN, Acadian Sea Plants and Green Science Technology, and 2 vineyards that collaborated, Arterra Canada, as well as Blessed Church. Thank you very much. And Perennial also played a very important role in this research. Thank you so much.
Great. Thank you, Dr. Sharif. That was a great presentation. So looking to see if there's some questions. And I see there is at least one question on the chat here. So it comes from Tim. It says, I'm going to read it out loud and can answer it. So thank you, Dr. Sharifi. Is there any work in Canada to look at the interaction of different rootstocks with your compost or VC/VCT treatments in relation to resiliency?
Very good questions. Unfortunately, I'm not aware of any research, but this is something important. So looking at the interaction of the rootstocks, we know that they're very important and they can play a very important role in the sustainability of the vineyard and resistance to the climate factors. So it's very important to think about this kind of research in different regions.
Thank you. There's also a comment on there. It says use of cover cropping favor anti-incidents in vineyards, but favors natural enemies. So that's great feedback there too. Do we have any other? Go ahead.
Absolutely, we have seen interactions with the natural enemies and attracting beneficial insects, as well as we saw an interaction of some of the cover crops with the powdery mildew. So we had lentils planting under the plant significantly reduced the incidence of the powdery mildew. So that interaction of the cover crops and diseases and pests, it's extremely important, and we're looking into that for the next 5 years.
Thank you. I don't know if we have any other questions, but feel free to put them onto the chat. I think, Dr. Sharifi, I think we'll stop here and move on to the next presenter, just keeping up with time, but thank you so much.
Thank you.
So as our next speaker, I'd like to introduce Dr. Harrison Wright, who is a researcher at the Agriculture and Agri-Food Canada in Kentville, located in Nova Scotia. His current work primarily focuses on plant physiology in both wine grapes and apples. Dr. Harrison will be giving us a presentation on climate change and the evolution of the Nova Scotia wine grape industry. So Dr. Wright, I see you on screen there, so over to you.
Thank you, Sergio. Just bring up my presentation.
We can see it, by the way.
You can see it. I seem to be the only one that can't see it. Just give me a second here. Okay. All right. Thank you, Sergio, for the introduction. It's a pleasure to be here to be able to talk to you all today. Sergio said that today I'm going to be talking about climate change and the evolution of the Nova Scotia wine grape industry. My name is Dr. Harrison Wright. I'm talking to you from the Kentville Research and Development Center, located here in Nova Scotia, Canada. So located in Canada, but for those of you not familiar with the geography of Canada, this is a map of Canada and Nova Scotia, or in Kentful, where the research center that I'm working out of is located, is hanging out here in the North Atlantic. So Nova Scotia, I mean, it's an interesting geography. It's, old mountain ranges and things like that are quite eroded. It was covered by a glacier during the last ice age and was pretty well scraped clean. And then when that glacier receded, it formed a number of very fertile valleys. One of the most prominent in Nova Scotia is the Annapolis Valley. It's where I grew up. This is a picture actually from the back of the farm I grew up, mostly known for apples, but been increasingly known for wine grapes. Annapolis Valley is about 15 kilometers wide, about 125 kilometers long, very fertile soils. So a quick outline, wine grape growing in Nova Scotia. I'll be talking about a brief history. Most of it's recent. It had a long start, and then it's really started to expand here in recent years. I'll also be talking about Nova Scotia, quite related, how the climate has changed here in Nova Scotia. And finally, evolution of wine grape climate risks. So I'll be talking about frost and freezes. They're something that I've studied personally, but there's a lot of other risks when it comes to climate change, obviously. So first of all, I want to mention this climate change. Good news. I want to make sure this is known that I don't consider this a climate change good news story. This is a story about how climate change is affecting what we grow here locally, but I mean, climate change is quite serious, introduced quite a few threats, but it's also a story of how regions are having to adapt. And the wine grape industry, and the birth of the wine grape industry here in Nova Scotia, which was historically very cool climate, but it's growing less so, is a story about how things are changing because of the changing climate. So wine grape growing in Nova Scotia, brief history. It's kind of an interesting history. So some of Canada's first cultivated vines were actually in Nova Scotia, of all places. These were brought, it was a very early European settlements. A lot of these settlers arrived in the 1600s and the 1700s. If you go back in the records, you can see accounts, sea captains, wealthy elite, talking about their attempts anyway to grow grapes in Nova Scotia. There's quite a few stories about early settlers getting here, and the first thing they do, because they come from places like France, and especially France, is seeing if they can grow grapes here. So accounts of such as this are in places like Bear River, Petite Riviere, to name a couple, where these early attempts at grapes were. Again, mostly I would not describe it as an industry, mostly just people trying to grow grapes. So for centuries, wine grapes remained what I would say the domain of backyard gardeners, hobbyists, and for the most part, the elite, people who could afford to do so. This was not a money-making venture by any means, but people trying to grow grapes. So that pretty well described wine grapes in Nova Scotia from the 1600s to 1800s. I should mention that there is not believed to be any indigenous grapes to Nova Scotia. Like I said, it was scraped clean by those glaciers. There was wild grapes locally, not too far away on that, what I call it, in Canada, just for Perry and other places like that, like only hundreds of kilometers away. But Nova Scotia is not believed to have indigenous grapes. So I did a project a number of years ago looking at endophytes, and evidence of wine grapes being grown here throughout the centuries. It was quite prominent. So a lot of old abandoned farmhouses in Nova Scotia. There's a history of agriculture here, and around these old farmhouses, you'd find evidence of everything they could grow. We're mostly known for apples, but almost every old abandoned farmhouse you have, or quite a few of them, you'd see remnants of grapes. These grapes would get in the woods and they just take off, and so you can find these wine grapes all over the place. And these were cultivars, not wild grapes. These are, I guess, what you describe as feral grapes. So old abandoned farmhouses, if you poked around, you'd find feral grapes from old attempts of trying to grow grapes. Other prominent places, this is what we call the French Mission locally. Some old historic sites, you'd often find grapes on it. The French Mission, it's believed to be the oldest building east of Quebec City in Canada. Again, there's not good records, but it's believed to have been built in 1699, an old stone house, and it's completely covered with wine grapes. But again, not an industry. Here's another example. There's another one. This one's in the Annapolis Valley here, not far from the research station, Prescott House. 1800s, again, evidence of growing grapes then. This was a prominent merchant from Halifax who had a second career as a horticulturist. Quite famous for a bunch of things, but he was also known for growing grapes. Again, short of an industry, more to impress his friends, but he was growing vinifera back in the 1800s here. Like along stone walls, or he also had set up some heated greenhouses to keep his vinifera alive through the Nova Scotia winter. So again, growing vinifera in Nova Scotia, even in the 1800s. Again, not an industry, but more of a hobby. So wine grape research. Again, there's the Nova Scotia Fruit Growers Association is a local association established in 1863. So a lot of history there. Not primarily known for growing apples, but again, even as far back as 1876, the Fruit Grower Association sent 14 varieties to the Philadelphia Centennial Expedition, which was in the U.S. So they had sent 14 varieties. There wasn't an industry here, but again, there was interest even that far back. This is actually an old picture of the research center where I work at today. It was one of the earlier research stations that was established in 1911, and about 2 years later, they started doing wine grape research, even though there was a lack of an industry. But there was always this interest, and I would say it was just a very low level of research activity on grapes. Predominantly, 99% of it was on apples, but even back then, they were doing work on grapes. Trying to see if an industry was possible. Here's a modern picture of the research center that I work out of here in Kentville, Nova Scotia. And here's an old picture. This is from the 60s or 70s, or probably the 70s, of some of the grape research trials. Again, these predate an actual industry here, what I would say a commercial industry. So this wine grape research that occurred here, it was, like I said, a bit of a low level, but it kind of continued for the better part of a century, or at least 80 years. And most of it was cultivar evaluation trials, things of that nature. They were just trying to get something to grow here that was wine grape. There was a lot of, I can't stress that there was a lot of interest in making a wine grape industry, even before it really seemed possible. This wine grape research kind of came to a head in around 1970. There was a big push to try and grow grapes here, a research push, I should say. There's a big trial. It began in 1962, which just so happened to be a relatively poor decade in terms of climate that was suitable for wine grapes. But it was published in 1970. This trial was, again, a cultivar evaluation trial. And it was conducted over multiple years across multiple sites. All the sites were within the large Annapolis Valley. It included multiple varieties, but the varieties, they knew we were at the limits here. It was a cool climate, everyone knew that. So they only tried to grow North American species, like really cold hardy species. And hybrids, things such as Concord, New York Muscat, Sybil, things of those nature. And after this big push to try and see if we could grow grapes here, again, I take this excerpt from the conclusion, which was the first line of the abstract in this paper. It's referring to Nova Scotia, in particular the Annapolis Valley, which is probably the most suitable in all of Nova Scotia, or one of them, as far as climate. And essentially, it says the successful growth of wine grape cultivars is not feasible. The primary reasons they cited was lack of heat. Wasn't warm enough to ripen grapes here. Even the really hardy hybrids or the really hardy North American species. They didn't even talk about vinifera. That was one of the questions. And so that things kind of died down for about a decade in terms of enthusiasm over wine grapes. This was a big initiative, and it kind of came up short in the researcher's mind. And that was that, at least for a little bit. But then about 15 years later, Nova Scotia wine grape industry I would say, was born. So late 1970s to present, we started seeing vineyards pop up. One of the first was Grand Prairie. It was in the late 1970s. Yost was amongst some of the early ones to take part in this industry, or to start this industry. By 1982, there was enough growers and people who are actually actively growing wine grapes and selling it that they established the Grape Growers Association of Nova Scotia. So something was changing here. And then what I would say is modern day. So now modern day, today, vinifera, more apt to be found around Mediterranean stuff, start to be planted in numbers, and they seem to be they're doing well at this point. Now they're doing really well. So things like Chardonnay, Pinot Noir, Riesling, what we refer to as some of the hardy vinifera that come from some of the more northern regions in in Europe. You know, they're now frequently planted in Nova Scotia. In fact, Chardonnay in particular is probably one of the more common grapes to be planted nowadays. So, and again, so in recent years, the industry has expanded greatly. The economic impact in Nova Scotia alone is estimated to be around 245 million. And in Canada, the impact is over 6 billion. So cool climate grapes, but it's really, especially Nova Scotia, expanded. So I guess I'll just overview. So in the 1600s, 1960s, I mean, there's this long simmering interest in wine grapes, hobbyists, enthusiasts. They only played around with North American selections, hybrids, and there was no commercial industry. It was just people like doing everything they could to try and grow grapes, mostly for their friends, but it was ultimately deemed to be too cold. 1970s to 1990s, wine grape industry was born, but only hardy North American selections and hybrids were grown. I mean, a few people dabbled with vinifera, but they soon switched over to hybrids, or usually after they lost their grapes. But then this started to evolve 1990s to the present. There's been rapid expansion in the wine grape industry, and new plantings are increasingly comprised of, you know, the less hardy vinifera species, the ones that need a warmer climate. So explaining how has this changed? Well, it's a climate change story, of course. So we've got data here from the research station. Again, the research station has been recording data here with calibrated sensors dating back to 1913. That's 2 years after the initiation of the station, but we have daily records going back to 1913. So over a century of daily mins and maxes every day from the research center, from the exact same site. So it's quite a historic data set. It's quite interesting to look at. So I'll just review what the wine grape region needs. You know, what do you need to be a wine grape region? So you need a mild winter temperature. I mean, we get winters here in Nova Scotia. You can't be too cold or you lose your buds on your vines, your fruiting buds. You need heat in the form of growing degree days, and you need a long growing season, long enough to ripen the fruit adequately. So this is, again, this historical data that comes out of Kemptville, Nova Scotia here. And we're just looking at the trends. And again, climate change, we know it's getting warmer. It really depends on where you are. I mean, how warm it's getting or how quickly. But when I look at our local data set, we can see over the last century or so, the temperature on average has increased about 1.9 degrees. Again, it's highly variable, but you see a definite upward trend that's highly significant. More dramatic is the minimum temperatures. This is a crucial metric for what we can grow here in terms of species. It's increased 5.9 degrees, you know, quite a bit more than the average temperature. So a lot of that average temperature is increasing because of the more mild minimums, daily minimums and the winter minimums. In growing degree days over that same time period, we've increased about 27%. Again, a little bit over the last century, you know, a significant increase in heat. So how about the growing season? How has it changed? So this is the spring frost timing. So in cool climates like we have in Nova Scotia, we define our growing season as the last spring frost is the beginning of the growing season, when your plants no longer get killed by those killer frosts. And the first fall frost is the end of your season. So for many crops, including grapes. So the spring frost, again, highly variable from year to year, but again, the trend is significant. You know, back in 1913, it used to be about May 26th, we'd receive our last killer spring frost. But today, that's May 8th. So on the front end of that growing season, we have an increase of 18 days here locally. In the fall frost, we used to get our first fall frost that shut down, you know, all green tissue around September 24th. And now that has now been pushed back to October 16th. So again, on the other end, we added 22 days. So in total, we've added on average, again, it's highly variable from year to year, but the trends are significant, over 40 days, you know, over a month more growing season. So that's quite a significant change, and it changes what we can grow here now. So lastly, I'm just going to talk about how this has changed. I mean, obviously we used to not be able to grow grapes here. Now we can grow grapes here in the form of a modern industry. So the evolution of some of the risks to growing grapes and how they've changed. So frost, that's a big deal here in Nova Scotia. 2018, again, that our industry has really just been around for the last few decades. So in 2018, we had a major frost on June 4th. This is a picture of someone's windshield. It's in French, but it says June 4th, 2018, and they have a hard frost in their windshield. That's not when we're supposed to be getting frost. And so on June 4th, we had at Kempfel here, it was highly variable. When you have a temperature inversion event, I mean, the temperatures are all over the place, but here in Kempfel was minus 1.87 on June 4th. That was the coldest June temperature on record. That record, goes back to 1913. And there's reports of far colder temperatures in the region. Like I heard reports of as cold as -8. Again, it depends on exactly where you were. If you're in a narrow valley or something of that nature, it would be colder. And this was the impact. So everything was just starting to grow. We had a lot of heat the week before. Things were growing nicely, and so it usually kills all those buds and all those shoots. Usually the base of the shoot might survive, but the tip, dies along with the meristem. And the fruit for that year, also very sensitive. The inflorescences here, they're killed in these kind of temperatures. So in general, Nova Scotia, the yield that year was down approximately 50%. Not everyone was hit equally. So some sites had no crop, and other sites fared better. But the number one question, again, because we're a relatively new industry, we were wondering, like, is this climate change? Is this a glimpse of things to come? Like, we're going to get more frost? Is the frost risk increasing? So to answer this question, we took what I would call our most precocious variety, or one of them, which is Lucy Coleman, a hybrid that breaks very early in the season, so it's very vulnerable to frosts. And we looked at the data and came up with a model that predicted the timing of bud break. Because the question with frost risk is, we know the frosts are coming later, but we're also wondering how you also have to look at the phenology of the grapes, how more heat is affecting the development of the vines. So if they're not in sync, you're gonna get a change in frost risk. Either it's going to get worse or it's going to get better. And again, this really depends on the region you are, or which way it's going to go. So we looked at a bunch of models, and we eventually went with what I call a simple forcing model, which just relies on heat. And we got a pretty tight model. It was 155 growing degree days at base 5 from starting at March 1st, and it was accurate to within plus or minus 3 days. So then we went back to our historical data set, and with our model, we looked at what would have been the bud break time for Lucy Coleman based on our historical weather data. So this is a graph of that. We can see they'll start breaking early and early with the increased heats. So we know that they're going to be breaking earlier with the heat, and then we compared that with actual date to the last frost. And the idea is you want your vines to break and to start growing, after the last frost. So you can see these trends were actually almost spot on. If you looked at the slopes of those two trends are almost identical, and sure enough, when we subtracted them, we see a straight line. This is the date versus the bud break, and we can see there's really been no change in the time between bud break and last frost. Again, we want that to be a positive number. We want the buds breaking after last frost. So based on our model, we see that there's actually been no change in frost risk. There's about an 8-day buffer for Lucy Coleman in the last frost, and that hasn't changed. So even though they're developing earlier, it seems as though at least the risk for frost, at least in this region, has not gotten worse. So another threat to grapes, Nova Scotia winter freezes. I mean, you see those trends that we've looked at. They're highly variable, but we still do get freezes. We know it's getting warmer. In the last couple of years, this has been a pertinent question because we've had a couple cold nights. In the last 3 years, 2 of those years, we had some temperatures in some places reach below -25, which is not good for vinifera, and even the less hardy hybrids. It's not very, it can impact those as well. So generally, when you get a winter freeze, if you open your buds, again, I know in some of the regions, people tuning in, they don't have to deal with this, but these cold temperatures, often kill your primary bud. And sometimes you'll see your secondary bud will hang on a little bit longer. They're usually a little bit more hardy. They're a little bit smaller, a little bit less water in them. If it gets too cold, it'll kill those as well. And for vinifera, most of those aren't very fruitful either. So it doesn't even help with certain cultivars. Some of the hybrids, those secondaries can be fruitful. So usually when we have one of these freeze events, you'll get bud damage, loss of crop, you'll get vascular damage, and you'll get a protracted bud break and vine collapse. So this is a good example after one of these freeze events. You see our hybrids in the back. They're more hardy. They have a nice canopy. But in the foreground, this is Chardonnay, and you'll see very sporadic shoots breaking after this winter freeze event. And you'll see quite a difference in when they break, and you'll see shoots coming from the latent buds on the trunk, which is not normal. That's a sign that those have been damaged. So again, just to look at the difference between hybrids and vinifera, we grow both now. We used to never grow vinifera, but now we do. We used to grow hybrids, and this is the reason. You can see the bud hardiness of the hybrids. This is our most popular hybrids across a number of sites. The bud hardiness, LT50 value, which is the temperature at which 50% of the buds would die, is about minus 26.9 across all our hybrids. I mean, it's variable, but that's the average. And the vinifera on average about 3 degrees less hardy, about minus 23. And that 3 degrees actually makes a big difference. But I'll just revisit the winter minimum historical temperatures we have here, keeping in mind those hardness values we had before. It used to be, approximately 100 years ago, on average, we'd get a minus 25.9 temperature each winter. And that has increased all the way to minus 19.9. So again, remember those hardness values. Now our average value is quite a bit below what those hardness values for the hybrids and vinifera are. But again, it's highly variable. So some years, if you grow those vinifera, you still do get winter damage. And, I should mention also, it's kind of interesting to see warm winters. Winters where you have temperatures, below minus 19, well below minus, or a warm winter, I would describe that as. In the first 80 years, we only had 2 winters like that. But things have really changed here, especially in the last 2 decades. So if you see in the last 20 years, nearly half the time our winters we don't even reach minus 19 that winter. So we went from having one every few decades to every other year when you have a winter that is quite warm, doesn't even reach minus 19. Although it is still possible to reach these cold temperatures that we've seen, but yeah, quite a shift. Again, better than averages is looking at the frequency. I would describe minus 22 as a slightly damaging winter, and you can see we used to get those almost 100% of the time at minus 22 or colder, and now it's only about 20% of the time. I would describe a minus 25 event as highly damaging to both vinifera and hybrids. And it used to be that would happen almost every other year, and now it only happens, very rarely. So one of my last slides. So is the risk of winter damage decreasing? I would say yes and no. I mean, our risk of really cold temperatures is definitely decreasing. We see how much our winters warm, but as we have warmer winters and especially warmer growing seasons, growers are pushing the envelope and growing less hardy varieties. So the effect of cold temperatures is decreasing, but because we're growing less hardy vines, they kind of balance each other out a little bit. So finally, I won't go into this, but we can see a reduction in risk just in those 2 things. Winter freezes and the risk of frost did not change, but things I'm not talking about, and I'm not a climatologist, is hurricanes. We get hit by those pretty frequently here in Nova Scotia. Declining snow cover is an issue. We can no longer count on them, that the snow is insulating. So, and we used to have snow throughout the winter, and now it comes and goes. Extreme rain events, thunderstorms, hail, these are all things that are questions, and disease and pest pressure. These are also things that are changing. That concludes my talk, and I appreciate everyone taking the time to listen. Thank you.
Thank you, Dr. Wright. Beautiful landscape, by the way. It's on my 2 vacation at in the near future, and you just convinced me with some of your pictures there. But
thank you.
Thank you for that. So yeah, great presentation. I think I did see we're a little bit tight for time here, but I think I do see some questions. So let's try to get to a few of them. I think the first one, I'll try to do my best at translating this. But I think basically it says, I see that the regression coefficients are quite low. Is there an explanation?
Yeah, I guess, I mean, this is classic weather data. I mean, the coefficients are very low. Again, these are not good prediction tools. It's really just looking at trends. The trends are high significant. You see they're all like p-values less than or 0.001. But yeah, I mean, there's so many factors that affect the weather, especially here in the maritime climate. Yeah, it is highly variable. So I guess I don't know if I have an explanation. It's really just the weather, and it seems to be especially prominent in the Maritimes. But the weather's highly variable. Not a good predictive tool, those trends, but it is indicative of there being trends, I guess.
Okay, thank you. And then the next one is there's a question here. It says, with this rapid expansion of new vineyards, what are some, or what do you see as new pest challenges in Nova Scotia in the past 5 to 10 years?
Of pest challenges. So so yeah, this is ongoing research as well. We're seeing viruses are becoming more prominent. Most of the viruses we see in Nova Scotia are believed to be being spread via propagation, but we're seeing glimpses of certain pests that we think are vectors. New instances in Nova Scotia. There's a grapeberry moth. Maybe we're thinking we might see it anytime. There's a whole list of pests that we've never seen before, and now we're starting to see, small pockets of it because of the warming temperatures. We used to not be able to grow vinifera here. Now we can, and we used to not have some of these pests, and now we're seeing that they're coming here mostly because of these warmer winters and the warmer seasons. They're coming too. Yeah.
Great, thank you. So thanks again, Dr. Wright. Thank you for that. I think we're going to move on with the presentation, but if anybody has any questions, I'm sure Dr. Wright wouldn't mind answering them via the chat. So feel free to still ask some of those questions. And so with that, I think I'll now pass the mic over to Dr. Tim Weidemer to introduce our next two speakers. So Tim, over to you.
Thank you very much, Sergio, and Dr. Wright. That's what we call job security. And I too enjoyed looking at those Nova Scotia views, and I've always had that on my list, so that does convince me as well. States. And so the first one is given by Dr. Lance Cato-Davidson, who is a research plant pathologist at the USDA Agricultural Research Service Grape Genetics Research Unit in Geneva, New York, which is co-located with Cornell Agritech. For the past 2 decades in this position, he has studied the genetics of grapevine interactions with fungal and oomycete pathogens, primarily focused on host resistance to grapevine powdery mildew. Since 2011, Dr. Cato-Davidson has co-led the USDA NIFA-funded VITAS Gen project, which provides technological innovations and centralized expertise to grape breeders across the US. Before joining the ARS, Lance received his Bachelor's of Science in Biochemistry at Kansas State University. He spent a year as a Fulbright scholar working on integrated intensive farming systems at the MS Swaminathan Research Foundation in Chennai, India, and then earned his PhD in plant pathology at Cornell University, where he focused on soil-borne viruses of wheat and other small grains. His presentation today is direct and indirect impacts of abiotic stresses on grapevine powdery mildew. Lance, please go ahead and take over the presentation.
Great, thanks so much, Tim. You see me and see my presentation okay?
Looks good.
Hear me? Okay, great, thanks so much. And I want to thank the organizers. It's a real honor to be able to talk today to you. And also want to give a special shout out to the interpreters for the job they're doing. We all love talking about our science, but I'm sure it's a real challenge to try to translate the complex terms and especially the species that we talk about. So the work I'm talking about today is quite different from what I normally talk about. I wanted to develop a talk related to climate change, of course. And so I pulled through a bunch of data that I developed over the past 2 decades with Dr. David Gadori, who is an epidemiologist working with me here at Cornell University. David just recently retired, so I want to mention him as a co-author of this presentation. So Harrison did an awesome job of providing examples of how climate is impacting grape production. And I think the data that we have in grapes shows a very compelling story of climate change. And it's one that people can relate to. I think most people like grapes in one fashion or another. And so it impacts them to see how the climate change is impacting grape production. And a really good example of this is how European grape production has changed over the past few decades. So in the upper right-hand corner here, you see the growing season temperature for Bordeaux, a region in France that grows grapes, has increased about 3 degrees C warmer temperatures. Steve Kappas mentioned up front that if the US grape production shifted 2 degrees C warmer, we'd lose about 56% of our production. So this is highly relevant scale of change. We also see that the harvest in the Alsace region is about 28 days earlier, about 4 weeks. And while this may sound good on some days, there's a lot more alcohol in the grapes that they're harvesting and a lot less acidity. But this is actually a very bad quality trait because when people are tasting wines, when they taste a higher alcohol wine, it comes across as hot or kind of burning, right? And when they taste a wine with low acidity, descriptors are commonly things like flabby, which are really not desirable. So a lot of negative things happening due to climate change in the grape industry. And I'm not an expert in climate change, but what I do know is that on a continental basis, the people in this room right now are all affected differently by climate change. So for my talk, I'm going to bore down to very specific abiotic factors that impact life and disease specifically. So, and just as a starting point, I think one of the central tenets of plant pathology is the concept of the disease triangle. And that is, in order for disease to occur, you must have a susceptible host, the presence of the pathogen, and a suitable environment for disease. And if you change any of these factors, you change the outcome of disease. And so in changing the environment, we definitely are impacting the disease triangle. And a concept that is as old as the concept of the disease triangle, but perhaps less often discussed, is a concept central to my talk today. And that is the difference between the direct effects and the indirect effects of environment on the pathogen. So this was mentioned on Tuesday during the wheat talks. Juan mentioned, he showed actually data for optimal growth optimal growth temperatures and conditions for a fungus. And here I show a similar thing for Gibberella, a pathogen, a fungal pathogen that infects both wheat and corn. And so if you just look at the fungal growth curve, you see that there's a broad range of temperatures that are conducive to growth in vitro by this fungus. And this overlaps quite well with disease on wheat and the optimums for disease on wheat by Gibberella. And this would be direct effects on the pathogen, right? So temperatures directly affecting fungal growth and disease expression on wheat. In contrast, if we look at corn, it's the cooler temperatures where you see more disease symptoms. And I consider this an indirect effect of temperature through the host on how severe disease is. And I'll show some examples of that today. So the pathosystem I'm going to talk about is grapevine powdery mildew. It's been brought up a couple times today. It's an economically very significant disease anywhere grapes are grown. A typical grape grower will spray 10 to 15 times a year for powdery mildew, and it makes up about 7% to 10% of production costs for grapes. So I'm going to talk about 3 different topics, 3 different biological aspects of powdery mildew growth and management. The first one is affected by winter temperatures. The second one is affected by springtime temperatures. And the third one is a concept of growing season control of disease. So I will dive right into these. And the first one, ontogenic resistance or age-related resistance. So the concept of ontogenic resistance is that fruit are highly susceptible to infection for a very defined period and then become essentially immune to later infection. So we can see that in the picture of the grape clusters here. We inoculated this grape variety at either 2 weeks post-bloom, 3 weeks post-bloom, or 4 weeks post-bloom, and then just allowed the disease to progress throughout the growing season. And hopefully it's very clear for you to see that we had very high levels of disease. The fruit was very susceptible early and became essentially immune later in the growing season. And that can be shown here by the relative susceptibility curve, a very sharp drop off in susceptibility as berries mature. We see the same trend for downy mildew and also for a fungus, black rot, Guignardia, though to less extreme extent for Guignardia. So this ontogenic resistance is impacted by the uneven phenology of grapevine. Okay, so let me just explain this a little bit. So in the northern climates, like if we think of Ohio shown here, New York, the upper Midwest, much of Canada, we get cold winters and a very synchronous development. So bud break is synchronous across the vines, across the vineyard. Flowering of the clusters along the vine is synchronous, and the individual flowers within a fruit cluster also synchronous. This is in contrast to warmer climates. We think of Australia as shown here, California, probably much of Mexico, where mild winters do not synchronize development. And so this will cause bloom to occur over a more expanded duration, which means that that period of ontogenic susceptibility is much longer than it would otherwise be if you had a synchronous development. So that is one way that I see climate change potentially impacting grapes and other crops, other perennial crops. The second topic I'm going to talk about is stress-induced disease resistance. So David had collected as an epidemiologist years of data tracking disease progression of powdery mildew and was surprised to find that the average temperatures, average daily temperatures, predicted powdery mildew epidemics should progress much more rapidly than they actually did. And it was only when he looked at the nighttime minimum temperatures that he had a good fit to the curve. And so we started exploring the biology of why nighttime minimum temperatures in the spring would be a good predictor of powdery mildew severity, disease progress. Before I dive into that biology, I want to mention that cool temperatures at night occur most places where grapes are grown, right? So it doesn't matter if you're a warm climate production region or cold climate production. This is generally an important topic for us. And in fact, the interval from bud break to bloom, almost anywhere you are in the world, you have about the same number of cool nights that this phenomenon is relevant to. So cold cider is an important phenomenon anywhere grapes are grown. The other thing I want to mention is, we think of ambient air temperatures, and that tends to be what we measure. But in the evening, the temperature of a leaf surface is actually cooler than the ambient air temperature. So if we're looking at temperatures that are lower than 10 degrees C, and we're thinking about just the ambient air temperature, it's actually colder than that on the leaf surface. It's colder than that in the environment that the pathogen is experiencing. So about a decade ago, a PhD student with David and I, Michelle Moyer, who's now a viticulturist at Washington State University, she did a pretty elegant experiment. So she inoculated powdery mildew onto susceptible grapevines and allowed a colony to grow for about 3 days. And that's the colonies that you see on the left-hand side of the figure. And then she cold treated them, 2 degrees C for 2 hours, or the top one, she didn't treat with cold, so zero time at 2 C. It was maintained at room temperature. And then she did a viability stain with FDA stain. And you can see that the one that was not cold treated, pretty much every cell is living. It looks like a very happy mycelium. And the one that was cold treated at the bottom, most of the cells died. There are very few cells that are still alive. And so it really knocked back disease progress. So this is a direct effect on the pathogen, right? The cold is actually actively killing the fungus. But we also found that if we treated the host in the absence of the pathogen, and then at some time later inoculated the pathogen onto the grapevines, we could detect what we call the cold stress-induced disease resistance. So if you just choose one of these leaf positions, let's say the second leaf position, the control grown at room temperature has a very high level of disease, and we get about a 20% reduction in disease severity if we treat the leaf with cold temperature for a couple hours. Now, this resistance is a transient resistance. It's not permanent. It lasts for about 12 to 36 hours. It starts about 12 hours after the cold is initiated, and it lasts until about 36 hours after the cold is initiated. So that was a small effect. This slide is focusing on what we call penetration success rate, which is just for a spore, how likely is it to infect the host and continue to grow to any extent. But then we can go on and look at how likely that spore is to continue on and be successful and make branched hyphae or start to form a mycelium. And we see that how extreme that temperature is actually predicts or impacts how successful the pathogen will be. So with a exposure to 2 degrees C, we see about an 80% reduction in the proportion of spores that are able to go on and make branched hyphae. So this is quite a strong effect of cold, indirectly expressed through the host, a highly susceptible grapevine becoming resistant, but temporarily. So we've gone on to look at this phenomenon on other hosts, and it occurs in pretty much any host that we've looked at so far. So we've looked at hops, we looked at strawberries, we've looked at Arabidopsis, which gives us some genetic tools to explore the basis of this. We've looked across the diversity of Vitis, and it occurs in all Vitis species, except for one, Vitis rupestris, that we are currently studying the genetics of why that species doesn't have cold stress-induced disease resistance. And I should mention, it's not just cold that triggers this temporary resistance. We see almost the exact same induction of resistance on susceptible varieties after exposure to heat. So 40 degrees C triggers a transient resistance in the host. Okay, the last thing I'm going to talk about is ultraviolet light, which I think is a pretty cool and impactful story. So about 30 years ago, scientists here at Cornell Agritech started playing around with UV, building a tractor-drawn implement like this and blasting grapes to see if they could get disease control. And from time to time, it was highly successful, but it was highly uncontrolled. And they ended up over-treating the grapes and getting russeted grapes. It looks like potatoes on the vine. And that's really an undesirable outcome. So we know that UV is a general biocide, right? Depending on dose, you can mutate DNA and kill many organisms, right? So stepping back to think about how ultraviolet light can be used to suppress disease, you think about the fact that the host and the pathogen have co-evolved with each other since long before the dinosaurs were around. And they've co-evolved in an environment of 24-hour day and night cycles, right? Cycles of light and dark. They've co-evolved with UV. And just like plants sense and use light, pathogens also sense, interpret, use light to direct their own development. And so in evolving in this scenario, and with a constant exposure to UV, because this fungus, powdery mildew, is almost wholly external to the host, so it's being bombarded with UV daily. powdery mildews and many other pathogens, evolved systems to repair DNA damage from UV. Now, one of the most effective such systems is a photolyase system. It's very well characterized in many different organisms. Photolyase repair systems use electron transport and are recharged by blue light. Now, blue light occurs in the daytime. It does not occur at nighttime. And also, these photolyase genes are shut off at night during darkness. So this kind of gives us the background for a eureka moment. Actually, a discovery that was made in Norway by our collaborators, and has since kind of grown into the project I'm going to show you. And that is by treating plants with nighttime UV, we can reduce the dose from what you saw of the vineyard potatoes in my first slide. We can reduce it by about 10% of the dose that we would need during daytime and get very good disease control. So what's that look like? Well, here I want to share with you, the doses that we've dialed in on for grapes. These might be different than the doses you would use on more sensitive plants like tomatoes. But in grapes, we can apply 200 joules per meter squared once or twice a week with implements that move about the normal speed of a tractor. And we get disease control that meets or exceed commercial fungicides. So this is a big success story. We've done this, we've repeated this result in many years and different places on the West Coast of the US, different countries. So this is a pretty well-known story by now about powdery mildew control by UV light. But one of our surprises was when we were doing those trials, we have an emerging disease in the Eastern US that's becoming extremely severe in the past 5 years, and that's sour rot. It's a disease that involves a really complex multi-organismal relationship of fruit flies or wasps transmitting yeasts into grapes, starting a fermentation, and that ethanol is converted to acetic acid by acetic acid bacteria. Sour rot, so you essentially are producing vinegar on the vine. It's very undesirable. And because it starts at about 15 bricks, very close to harvest, there are not many tools in the toolbox for managing sour rot. And so one of the tools in the toolbox is oxidate. And we were very pleased to see that UVC actually reduced sour rot disease by about 75%, which better than oxidate, one of those only tools. And we've repeated this now 3 years in a row. So I think it's going to be a really important new tool, probably in the Eastern US, more important to use for sour rot than to use for powdery mildew. Another surprise, Jan Nijrup, who's the director of our experiment station and an entomologist, found that UV suppressed spider mite populations. So this is the two-spotted spider mite.
And you got about 1 minute.
Okay, great, thanks. I will zoom through this, no problem. In addition, I think this is a beautiful picture. We found in the lab that UV reduces downy mildew severity in the lab. And I think you can clearly see the white fuzz here at low or no treatments and very good control. But we were very surprised in a high disease pressure year to find almost 100% yield loss even when we treated fruit with UV. So it's not a cure-all, but it's pretty impressive. And we've seen, David's worked with others to show that it works against the bacterium Erwinia on fire blight, Cercospora on beets, apple scab, basal downy mildew, lots of different pests and pathogens. It's effective. I, in my research vineyard, use a simple lawn tractor and essentially a homemade array to pull the UV through, but there's this fun group, Saga Robotics out of Norway, that's developed a UV robot. And so just, I hope that I've given some examples of how how abiotic factors impact disease both directly and indirectly, how we can predict these effects, even though with imperfect accuracy, and how with knowledge we can harness the effects for disease management. So I want to give a final shout out to David Gadori, with whom I've co-led a really great lab group for the past 20 years. Thanks, Tim.
Thank you, Lance. And I did put a question in the chat, and there may be others. Thank you for that very interesting and fascinating presentation. Unfortunately, because of time, you can just answer my question there in the chat.
Will do.
Or something. So now we come to our last speaker of the day. It's my pleasure to introduce Dr. Lindsay Burbank, who received a bachelor's degree in pathobiology and veterinary science with a minor in molecular biology from the University of Connecticut. From there, she went on to receive a PhD in plant pathology and microbiology from the University of California, Riverside. Dr. Burbank joined the USDA Agricultural Research Service in 2014 as a postdoctoral associate at the San Joaquin Valley Agricultural Sciences Center in Parlier, California, researching virulence mechanisms of Xylella fastidiosa in grapevine. In 2016, Dr. Burbank joined the Crop Diseases, Pests, and Genetics Research Unit in Parlier as a research plant pathologist. Her current research is focused on detection and management of Xylella fastidiosa in grapevine and other host plants, as well as bacterial symbionts of insect pests, including vine mealybug. And Dr. Burbank's presentation this afternoon is temperature influence on Pierce's disease of grapevine, hot summers, warm winters, and increasing disease severity. Lindsay?
All right, thank you. Can you see the slides all right?
Yep, looks perfect.
Okay, great. So I'm going to talk about the climate influence on Pierce's disease. And because there's such a large body of research on this disease out of California over the last more than 20 years, I wanted to just make this a general overview of what we know about how Pierce's disease will be impacted by climate factors such as temperature and also drought stress as well. So some of this is directly related to my own research, but I'm also going to be talking about some other work from colleagues at the USDA, as well as the University of California. So, my slides advance. Why that is. Okay, there we go. So just to very briefly introduce Pierce's disease, this is a disease caused by the bacterial pathogen Xylella fastidiosa, and it causes leaf scorching on Vitis vinifera. Most European grapevine varieties are very susceptible to it. You can see the leaf scorching on the leaves, and then it always has this very characteristic colored margin around the scorching symptoms, which can either be yellow or red, depending on the cultivar. And then as the disease progresses farther, you get symptoms such as shriveling of the fruit, discoloration of the vines, and eventually it will kill the vines over time. And it doesn't necessarily kill the vines in a single season, but since grape is a perennial crop, it's important to manage it so it doesn't continue into the next season. So this, again, is a bacterial pathogen, and Xylella fastidiosa colonizes a very specific part of the plant, which is the xylem tissue. So you can see in this microscopy image, all of the purple false colored bacterial cells within the xylem vessels of an infected plant And you can see just how many bacterial cells there are and how severe this infection can be. And this is one of the things that disrupts the water flow within the plant and causes the scorching symptom. So because this is a pathogen that is found only in the xylem vessels of plants, in order to be transmitted from one plant to another, it relies on insect vectors. And so we have here two of the insect vectors that are important for transmission of Xylella in California, the glassy wing sharpshooter and the blue-green sharpshooter. But it's really a very general mechanism of transmission. So you can have many different insect species that will transmit this pathogen. And pretty much any insect that can feed on xylem sap has the potential to be a vector for this bacterial disease. So I'm going to talk about the ways that climate can influence Pierce's disease. And I'm going to start with the disease pathology in plants and how this is going to be impacted by both the summer temperatures and the winter temperatures, as well as by drought stress. So to start with, we know that heat during the growing season can accelerate the disease symptoms of Pierce's disease. And so this comes from some work out of the University of California, Berkeley, from Rodrigo Almeida's group. And they looked at vines that were inoculated in a greenhouse and then held at either a higher temperature around 32 degrees or a lower temperature around 21 degrees. And you can see over time the percentage of plants that have an infectious dose of the pathogen. So they test for the bacterial populations in the plant. This will increase much faster when you have the plants held at a higher temperature. And at the bottom, the same is true for development of symptoms. If you have plants held at a higher temperature, you get faster development of symptoms. So there's a very direct connection between heat during the growing period and progression of Pierce's disease. In addition to heat during the summer, we also have to think about what the winter temperatures are going to be. And one thing we know from a history of research on Xylella fastidiosa is that the pathogen can actually be eliminated from infected plants over the winter under certain conditions. And this will allow the vines to recover the next growing season. So it's not only important to look at progression of the disease, but also what is the likelihood that vines will recover from the infection. And this comes from some work in the 1990s and early 2000s from the University of California, Berkeley. And they looked at what was the percentage of vines that would recover in relation to time of inoculation during the season. So what they found was that, at least in Northern California, where they did these studies, vines that were inoculated later in the season were much more likely to recover than vines that were inoculated early in the season. So vines that are inoculated early in the season, they have time to have a chronic infection established, and they're much more likely to remain infected into the next growing season. So in addition to time of inoculation, one of the other things that's very important when you're looking at what is the rate of recovery of vines is what are the winter conditions that these plants are going to experience. So this is based on some greenhouse experiments looking at number of chill hours and rates of vine recovery. And so the plants experience a certain number of chill hours, which is cumulative hours below 7 degrees Celsius. And then we looked at how many of these plants would recover. And if you put this in the context of different locations within the state of California, Temecula is down in the very southern part of the state. And here they don't experience very many chill hours during the winter. And so plants that are subjected to a treatment with that number of chill hours are not very likely to recover. But if you move up north throughout the state of California, all the way up to Northern California, the plants are going to experience more chill hours, and you're going to have a higher chance of vines recovering over the winter. And so this is based on experiments using Chardonnay inoculated in greenhouses. And I mention this because it's also important what cultivar you're talking about, because cultivars that are more susceptible are less likely to recover as well. So it's a factor of cultivar in addition to the conditions that are experienced. So if we put that together and we look at both the summer temperatures and the winter temperatures, you can get an approximate idea of what the risk of severe Pierce's disease outbreaks will be in different regions of California. So again, down in the southern part of the state, this area has very hot summers. So high summer degree days accumulated during the growing season, but they have also low chill hours during the winter. And so these areas are likely to be the highest risk because you're going to get faster disease progression during the summer, and you're going to get less recovery over the winter. And then if you look at areas such as the Central Valley, where I'm located, we still have very hot summers. And so you're likely to get fast disease progression, but we have colder winters than Southern California. So there's more of a chance that some vines could recover over the winter. And then an important area up in Northern California, this is the Napa-Sonoma region, where they produce a lot of very high value wine grapes. And this area has very mild weather during the summer. It's not nearly as hot. And it also has colder weather in the winter. And so they have more chilling hours, they have less heat during the summer. And right now they're at a relatively lower risk for severe Pierce's disease because of those factors. But you can see how this might change if the temperatures shift, right? So these areas up here that are now at low risk might be at a higher risk in the future. And it's important to consider this in our management strategies, that we might need to be more proactive about management in areas where we previously didn't have to worry as much. And so in addition to temperature, water scarcity is also a big problem in California. And right now, the Western United States is in several years of severe drought. But even if you look at the historic rainfall in the state of California, you can see that up in the northern regions along the coast and also in the mountains, you have a lot more rainfall in general compared with down in the Central Valley and then down in Southern California. So these areas down in the south where we have very hot summers, we also have a lot less water. And grapes grown in these regions are irrigated, but more and more as time goes by, water is becoming scarce in California, and there's less water to use for everything, including for irrigating our crops. And so people are trying to work with less and less water as time goes on. And when you look at the pathology of Xylella fastidiosa, it's very directly connected to water status of the plant, because the pathogen is growing in the xylem vessels, which are the water conducting tissue of the plant. So you've got bacteria colonizing the xylem vessels. And in addition to just bacteria in the xylem vessels, the plant will also launch a response to the bacteria. The plants will form what's called tyloses, or these obstructions in the xylem, in an attempt to control spread of the pathogen. But this can also obstruct water if it gets out of control. So together, both the bacterium and the plant response ends up restricting water flow within the plant. And this is what contributes to the scorching symptoms that you see with Pierce's disease. And so this is from a study from the University of California, where they looked at a combination of Xylella fastidiosa infection as well as drought stress. And what they saw here was that if you have plants that are experiencing drought as well as infection, you have more symptoms develop than if you only had the pathogen alone. And in addition to that, when they looked at gene expression patterns of the plant, what they saw was that gene expression patterns caused by infection in some ways were very similar to what you would see caused by drought stress. So the two stresses combined can have a greater impact on the grapevine than just the pathogen alone. And so that's something we're going to have to consider as we deal with more drought in grape growing areas. So those are some of the ways that the pathology of Pierce's disease is impacted by climate, both by summer and winter temperatures, as well as drought stress. And now I'm going to talk a little bit about the insect vectors and how this component of disease is also influenced by climate. So as I mentioned, this is a pathogen that's transmitted by insects. It's a general mechanism. It can be transmitted by multiple different species of insects. I'm going to talk about the glassy-winged sharpshooter and several studies that have been done on the glassy-winged sharpshooter, but this is only a small subset of the research that has been done on vectors of Xylella. So some of the ways that climate can influence the vectors has to do with both the vector populations, the vector feeding behaviors, and then also survival and mortality of the vectors throughout the course of the year. So to start with, this is a study from my colleague Rodrigo Krugner, as well as others at the University of California, Riverside. And they were looking at deficit irrigation and how this impacts populations of glassy-winged sharpshooter in citrus orchards. And citrus orchards are important in the context of Pierce's disease because in many parts of California, you can have citrus growing right next to a vineyard. So in this photo on the side, it's looking out of a citrus orchard, and directly across the street is a table grape vineyard. So often you have these plants close together, and the insects can go back and forth. They don't have very much preference for what they feed on. So controlling the vector populations in the adjacent crops is an important part of controlling Pierce's disease. And what they found in this study is that if you look at citrus orchards, which are experiencing deficit irrigation, so at either 80% or 60% of the crop evapotranspiration rate, compared with 100% or no water stress, they found that at 60% irrigation, you actually had a reduction in the glassy-winged sharpshooter population throughout the season in the citrus orchards. So this seems like maybe not such a bad thing if more drought might actually reduce the vector population. And then to go down to a more specific level, this is another study from my colleagues here at the USDA, Rodrigo Krugner and Elaine Backus. And here they're looking at the actual feeding of the insects on plants and how the feeding behaviors could be influenced by drought. And they use a technique called EPG, where you have an insect attached to an electrode. And then when the insect feeds on the plant, it creates a complete electrical circuit through the insect and through the plant. And you can look at the patterns of the current of this electrical circuit, and you can identify the behaviors of the insect feeding on the plant. And so what they found was that when they put the insects on plants experiencing water stress, they had a reduction in both the number of events, of feeding events of the insect, and also the duration of feeding. So the insects seem to prefer to feed on well-watered plants, and they don't feed as much or for as long on deficit irrigated plants. So the vector feeding and the vector population, these things are only part of the equation here, because at the end of the day, we care about how well the vectors are transmitting the pathogen. And so this is a study from the University of California, Berkeley, from Rodrigo Almeida's group, where they're actually looking at acquisition of Xylella fastidiosa from plants that are under water stress. And they had insects, glassy-winged sharpshooter, held on infected water-stressed plants and then transferred to test plants to see how many of the insects acquired the pathogen and then were able to transmit the pathogen. And what they found was that if you look at from low to moderate water stress, you get a slight increase in the acquisition efficiency of the insects. So they're able to acquire the pathogen slightly more efficiently from the drought-stressed plants. But it gets a little bit more complicated than that, because in the same study, they also looked at a choice test where they have the insects caged with the drought-stressed infected plants at the same time as the test plants. And they looked at also a more severe water deficit in this case. And what they saw was that if you move towards a more severe water deficit in the plants, and the insects have a choice on where they feed, you actually get a reduction in transmission overall. So it seems like this story is a little bit complicated. Low to moderate water stress may increase the efficiency of acquisition, but looking at transmission overall under severe water deficit, you actually have less transmission. And so in addition to drought, I'm also just very quickly going to talk about temperature and the insect vectors. So this is the study from the University of California, Riverside, and they looked at feeding of glassy-winged sharpshooter under different temperatures. And you get an increase in feeding as the temperature goes up. And then down around 10 degrees Celsius, it's too cold for the insects to feed, and they stop feeding altogether. Now, they also created a model of this using cooling degree hours as a measurement of cold temperature. And they modeled what the population mortality of the insects would be at longer and longer periods of low temperature where they're not able to feed. And there is, of course, as the insects are not able to feed for longer periods of time, an increase in the percentage of the population that dies. And so when they looked at this in the context of winter temperatures in California, so specifically in Riverside, which is in Southern California, they predicted that somewhere between around 80 to 99% of the population of glassy-winged sharpshooter would be expected to die off over the winter. And it might seem like this isn't really a large difference, but if you look at it the other way, it's the difference between 1% of the population and almost 20% of the population of insects surviving to reproduce the next spring. So if you have a large insect population, this could definitely affect your population in the next season, and it could impact overall spread of the pathogens. So with that, I'm going to end. I think I'm probably close to out of time, but I just wanted to go through some of these ways that the insect transmission can also be impacted by climate, and to demonstrate how complex these interactions can really be, and how we're really going to have to consider all of these different factors to see how disease dynamics will change over time. And since I am the last talk of the day, I wanted to try and end on a positive note in some way. We've spent a lot of time talking about how disease will get worse. But I think the positive note, as everyone else has also talked about today, is how much information we do know about how these diseases will be impacted by climate. And so it's just a matter of using this information going forward in our further disease management. So thank you, and I can take any questions if I have time.
Thank you, Lindsay. We do have one question in the chat, and so I'm going to ask that so that it can be asked, because we do have maybe 1 minute. I know Dr. Lavalier is not going to talk too long, hopefully. Sorry, Jean-Charles. Anyway, water stress reduces the incidence of Pierce disease vectors, but what is the effect on plant growth?
Yeah, so that's a very good question. The level of water stress that they're talking about in a lot of these studies does not significantly impact the growth of grapevines. In general, they're not a crop that needs a huge amount of water. And I don't have a lot of information to give you as far as how much drought stress will start to impact the crop value, because that would be the important part. But in general, grapes are relatively tolerant to little water.
Great, thank you very much. So yeah, if you can just stop sharing your screen. And so that does conclude our presentations for today. You know, one of the advantages of these virtual webinars is we can open it up to a wider audience and have more people participate. One of the downsides, of course, is not having that direct interaction to establish collaboration. So I encourage everyone, the attendees, the participants, you know, to continue the dialogue together. You know, you gave some great presentations and overviews of the research and the problems. There's a lot of common problems, and so I encourage you to, you know, reach out either to myself or Sergio or Jose, if to help make those connections. We'll be happy to do that. And so at this time, I'm going to turn over for the final remarks. And I just want to thank again all the presenters and all the attendees. And now I'm going to turn over the remarks to Dr. Jean-Charles Lavalier, who's the Executive Secretariat of Prosa Norte and is stationed in Ottawa, Canada. Jean-Charles, please close us out.
Fantastic, Tim. Great, amazing session. I've learned a lot. I wanted to actually comment immediately to Lindsay, because I was thinking, okay, those chilling hours, principle, like it's a great concept. I'm going to use that with my kids. I'm going to tell them, you know, Dad's having a chilling hour while he's sipping some California wine, and transmit that information to the kids. You know, like anyway. So now, amazing session. I'm the executive secretary for Prosa Norte. As you know, it's a network of scientists from Canadian government, USDA-ARS, CNIFAP in Mexico. We host various webinars throughout the year. We had one on wheat 2 days ago. Similar to this, the sessions themselves, we often get this question: are they recorded? Yes. Will they be posted online? Yes. And so we're really, I've been enjoying the questions too. Very, very dynamic, lots of momentum. The grapes are a sensitive crop. I mean, this is a very important several $1 billion in terms of industry, wine across the world. And they're affected by diseases, they're affected by droughts. We're getting hotter summers, we're getting warmer winters, and you're seeing behavior changes. I'm an agronomist. I don't tend to work a lot on plant science, but I work more with farmers, and they really appreciate your work. I mean, it's very helpful to them to adjust their agriculture practices. And you can see where the wine maps, the grapes are expanding worldwide. There's greater demand too. And in terms of behaviors, and when I work with grape farmers and growers, you tend to see like they tend to seek higher ground now. They tend to shift away from the south to the north. So they're kind of curtailing the sun, if you want to call it that. They're trying to avoid over ripening of the fruit, and you can see different changes too in the varieties in grapes. And so a lot of behavioral changes, and plant health science really informs those decisions at the business level, and even in countries as an industry. Your work is very important. So my thanks to you. My thanks to everyone in the task force for putting an amazing agenda. My thanks to the speakers, excellent presentations. Thanks to the interpreters, thanks to my team here in Ottawa, the logistics behind, and team in Costa Rica, headquarters in IICA. I wanted to just extend one more thanks to President Dr. Kapis, to Tim as the head of the task force. Great job once again. And we do this every year. And I'm thinking now we should do this more often. There's so many commodities, and there's so much interest in climate change, and we're getting ready for COP27 in Egypt. and we're doing an agriculture in the Americas pavilion there, our first ever, and just getting a lot of traction. So really congratulations to everyone. Well done. And I would say santé in French, you know, when you drink, it's to your health. My thanks again. Congratulations. Take care, everybody. Bye-bye.